Transport of substances in a plant, AQA biology. Ketones, symplastic pathway, travels through the cytoplasm, apoptose way, travels through the cell wall, root pressure, the movement of ions by active transport, forces water up by a few centimeters, podometer, used to measure transpiration rate, translocation, the movement of sugars through the phloem. Transport of water in the xylem. What does the xylem transport? Well, it transports both water and ions. And the process that it does this by is transpiration. So how does it actually occur? Well, water enters the roots via osmosis and active transport for ions. And the pathways for water in the roots is the symplastic pathway, which is when water travels through the cytoplasm by connecting cells called plasmodesmata, and these have little holes which allow the movement of water. And the apoptose pathway, which is where water moves through fluid-filled spaces of cellulose through the cell wall. And due to the cell wall's thick depth, water diffuses easily and it controls the uptake of water from the plant. Mineral ion importance. Well, it helps to establish a lower water potential so the plant can take in more water. The movement of water through the stomata. The stomata is open, and as carbon dioxide enters, water leaves due to the opening. And this decreases water potential, which causes, think of it like a straw. So you have your straw in your drink. And let's get your drink. This is a very poor cup, but we're using our imagination here. Here we are. Alright, so you have your water. So as you suck on the straw, as more water is leaving the straw, you're sucking more up. So as more leaves this area, more gets drawn up here. And that's the same for transpiration. Factors affecting transpiration rate. Wind, humidity, temperature, and light intensity. If it's windy, then that means more water is going to leave the plant because an equilibrium isn't established because the conditions keep on changing. If it's a high humidity, then more water may enter the plant or less water may leave. At high temperature, you're drying out the water in the plant, ergo needing to pull more water up by the roots. Light intensity can also have an effect as you use more water due to there's excess light causing more heat and such. Structures of the xylem. It consists of cell wall, not the cytoplasm. They're dead cells, and cells are covered in lignin, which is a waterproof substance. And the evidence for cohesion theory. Well, at different times of the day, you have different diameters of the tree due to the suction of the tree being bigger or smaller at different times. So at the highest peak of the day, you're going to have more water being drawn up because you have more evaporation at the least. So if more water is being drawn up, that means the diameter of the tree is going to be pulled in. However, at night time when there's no photosynthesis going on, the diameter is going to be bigger because less water is being drawn up. And due to its cohesion theory, the water won't pull on the sides of the xylem. So that's the reason why sometimes you see people like hugging trees to see if the diameter is bigger or smaller at certain times of the day. Or you can use radioactive tracers, whereby not radioactive tracers, sorry, that's phloem. By using tracers, like for instance, when you have celery, if you were to put red dye into water and then put celery in it, you can literally see the water going up the xylem because of the change in color. It makes it more apparent. Transport of substances in the phloem. Phloem transports sugar by translocation. Now, what's the structure of the phloem? Well, the phloem is made up of sieve tube elements, And they have holes to form sieve plates. They have a companion cell for metabolic support, which allows all of the things that cells need to survive to be produced in excess so it can be passed along to the sieve tube element. So the sieve tube element has more space for the big vacuole, so it can transport as much sap as possible. And the structure makes it easier for the flow of sap. Now, what is the movement of the solute in the phloem? Well, active transport loads the solutes from the companion cell into the sieve tube element, which, due to this solute, it decreases the water potential. So, due to this, water moves in from the xylem into the phloem, which results in a high hydrostatic pressure. At the bottom, solutes leave, and due to this, it increases water potential. So, water now leaves at the bottom of this sieve tube column. Due to it leaving, it creates a high pressure at the top of the plant, causing the solutes to travel downwards. So let me draw a quick diagram so you can see what I'm talking about. So you have your sieve tube elements, which are like this. I can't even see this color so well. Okay. So you have your sieve tube elements. You have your xylem, which is, keep in mind, xylem and phloem are two different things. And then you're going to have your companion cell. So the solutes load from the companion cell, creating a low water potential, which causes water to move in to the sieve tube element. Keep in mind there's little holes that the solutes can travel down. So because of this, now a normal water potential gradient has been established, because remember the solutes help lower water potential. Now at the bottom, there is there's less solutes in this cell than there is in this cell. This isn't a companion cell. Let me change the color of this actually. Let's say this is a normal body cell. Let's say this is, I don't know, not a body cell. This is a normal cell, alright? So because there's more solutes in this area, sugar, in this area than there is in this area, the solutes leave. And as they leave, it creates a high water potential because you don't need all of that excess water. So the water moves back into the xylem, which means now this area has a high water pressure, so it moves downwards and the cycle continues. Hope that made sense. And the evidence is basically when the phloem is cut, sap oozes out showing the pressure gradient, or you could add radioactive traces from carbon dioxide. So once carbon dioxide is added and it photosynthesizes and such, and sucrose is made, which is the sap basically, you can see using a fluorescent screen how much of that radioactive tracer is being used and how it's moved throughout the plant. Now that's all there is for transport of substances in the plants, and the next video will be about cardiovascular diseases. So if you like this video, then please like and subscribe, and I'll see you in the next one.